Hey everyone, welcome to the 2DCC webinar. Today's speaker is Dr. Hebin Lee from Florida International University. And Dr. Lee, you are good to go ahead and get started. Thank you.
All right, thank you. Thank you for the introduction. And I thank the organizers for the invitation. And it's a pleasure to share some of our work on multidimensional coherent spectroscopy. And I will also speak about a few examples of using this technique to study many-body dynamics in atomic system as well as in 2D materials. So before I get started, just a brief introduction of my group. So the research in my group mainly focuses on the field of AMO physics. So we spend a lot of time developing techniques and tools in ultrafast spectroscopy and quantum optics. And then we use those tools and techniques to study first many-body physics in atomic systems in both atomic vapors as well as cold atom systems. And then we also use those tools to study dynamics, carrier dynamics in different materials like color centers, 2D materials, and perovskite materials. And of course, the work is all done by a group of postdoc and students in the lab. And this is a picture of some of the members in the lab that we took just a few weeks ago. And I also would like to acknowledge the funding support from different agencies. So for this talk, I will first start with a very brief introduction of optical multidimensional coherent spectroscopy. What is it, and how to do that experimentally. And then I will give you a few examples in a very simple atomic system in rubidium or potassium vapors and cold atoms. And then I will move on to give you a few examples of this technique being used to study 2D materials. Okay, so what is multidimensional coherent spectroscopy? First, I would like to explain what I mean by 2D or even 3D. So the dimension here of a spectrum refers to the number of frequency axes. For example, in conventional 1D spectroscopy, we normally plot the signal, which can be, for example, absorption or PL, as a one-dimensional trace as a function of the frequency, wavelengths, wave number, whatever unit you use in your field. So for example, what I'm showing here is an absorption spectrum of a potassium vapor. And so this spectrum has just one frequency axis. But if you don't know anything about the sample, this may actually give you some ambiguity because you could be looking at just one species of atoms with two different transitions, or you could be looking at two different species of atoms, but that gives you two absorption peaks as well. So sometimes it's not really clear what the picture actually is if you have only a one-dimensional spectrum. However, if we have another dimension such that we can look from another angle. So now in this case, you can find out actually there are two peaks hiding behind each peak in a one-dimensional spectrum. Now we can use contour lines to represent the spectrum, and this is the so-called a 2D spectrum. However, sometimes this is still not a full picture. If we add a third dimension, then you will see in a three-dimensional space, you will see even more features that you wouldn't be able to see in a one-dimensional or even two-dimensional spectrum. And moreover, when we perform this experiment, the data were taken in a coherent fashion. What that means is that all of the excitations are actually coherent. Therefore, the the three dimensions, the frequencies in the three dimensions are actually correlated. So you sort of have a correlation spectrum. So what I just showed you, this is not just an animation. This is actually an animation based on the real data we took on a potassium vapor. And if you don't understand what's behind the data here, that's okay. I will explain a little more in a while. Okay, so this idea of 2D spectroscopy is not new in optics. The concept of 2D spectroscopy or 2D Fourier transform spectroscopy originally came from nuclear magnetic resonance, NMR, where RF pulses are used to excite nuclear spins, and then you can record the response of the sample as a function of the time delays between different pulses, and afterwards a Fourier transform will generate a 2D spectrum like that. And this technique has been now commercialized. People routinely use this technique to study, for example, big molecules, proteins, and DNAs, and the idea is similar in optical multidimensional coherent spectroscopy. And Tamimura and Shao-Mokumil proposed in 1990s that we can actually use optic pulses to do this experiment similar to AMR. But actually is quite different in doing this AMR experiment in the optical region. There's a lot of technical difficulties associated with much shorter wavelengths in the optical region, but it took a while. But by now, the field has pretty well established, and this technique has been used to study a really broad range of different materials. And also this technique has been widely used in the physical chemistry community. However, it was not well known in the physics community, even though there are already a lot of applications in the physics and material science. Therefore, we recently wrote a book which actually gave an overview of this technique and what potential applications that we have in physics and material research. Okay, so what is 2D spectroscopy? So there are two keywords. First, Fourier transform, and then 2D. So I would just use this cartoon to explain the basic idea of this. So first, let's consider Fourier transform spectroscopy. Now, what if so what do we do if I have this tuning fork right here and I ask you to identify the natural frequency or frequencies of this tuning fork? Now, the simplest way to do this is to hit it with a hammer, and then you record the sound over time, right? And then what you can do, well, I'm oversimplifying this, so this is only a single frequency right here. So now you take a Fourier transform of this temporal signal, and then that will give you a signal in the frequency domain, and that will tell you the natural frequency or frequencies of this tuning fork. Right, so Fourier transform spectroscopy is an approach to measure the frequency response of a system by applying an impulse excitation and then recording the time evolution and then taking a Fourier transform. Now, this to demonstrate 2D spectroscopy, let's repeat this experiment in a slightly different way. Now, instead of using a hammer, now we're going to use a speaker. The difference is that the frequency of the speaker can now be tuned or varied. Now, by using that, we are able to correlate the sound signal of the fork with the speaker frequency. That is the excitation frequency. We can think of the y-axis is the speaker frequency or the excitation frequency, and then the x-axis is the fork frequency or the emission frequency. Right, so this will give you one peak that is on a 2D plane with the same absorption frequency and the emission frequency. Therefore, this peak is going to be on the diagonal line. Now, of course, if your system is this simple, you don't really need to do that. There's nothing interesting about it. But if you have now another tuning fork, then what you're going to have is that you're going to have another peak that's also on the diagonal line. Still, that's not that interesting. So now what's going to be a little more interesting is that if now you have a weak coupling between the two tuning forks, so that the energy can be transferred between the two tuning forks. In this case, then you will have two cross peaks that are not on the diagonal line. So those peaks are actually associated with the coupling and the energy transfer between these two tuning forks, right? So so that's the basic idea of 2D spectroscopy. And the way we're going to do this is that we're going to do this in the optical region. Therefore, we actually use a sequence of femtosecond laser pulses to excite the sample and then generate a nonlinear signal. And then we record the nonlinear signal as a function of the time delays between the excitation pulses. Now, for example, if we are looking at a simple homogeneous two-level system, this this is what the time domain signal looks like. So basically, this is just an exponential decay in the tau direction and an exponential decay in the t direction, which is expected for a simple two-level system. Now, what's interesting is that now if you do a Fourier transfer of that, the magic happens. So now you actually have a single star shape like peak on the diagonal line, and that's the simplest 2D spectrum that you can get for a simple two-level system. Now, in order to actually implement this in the experiment, there are a few requirements. That is, the excitation needs to be coherent, which requires all of the pulses to be phase locked. That is, you have to very precisely control the delay between all of the pulses, and any fluctuation needs to be either compensated afterwards or actively stabilized in order to have the phase lock between of the pulses. So there are different implementations of this experiment. In our lab, we have two different ways. So I'm not going into all of the details right here just to show. So the first implementation is in this box geometry, so that the nonlinear response of the sample comes out in a direction which is not in the direction of the excitation pulses. That actually is an advantage, because then you don't have the background noise from the excitation pulses. So another implementation is the so-called collinear setup, where we actually combine all of the four pulses back into just the one beam, and then all of the four pulses are actually modulated by ALMs at different frequencies. Therefore, effectively, each pulse is frequency tagged, and then that's how we actually demodulate the signal afterwards by mixing the frequencies, right? So the advantage of this setup is that now you can actually combine all of the pulses into one beam. Therefore, you can focus it really tight, especially you can actually couple them into a microscope. Therefore, you can look at the samples that are really small. Okay, and this is how it looks like in the lab. Here, this is a laser system. This is a 2D spectroscopy setup. And here, right now, we have a cryostack for solid state samples, and there's some spectrometers on the side. Okay, so that was the basic introduction for 2D spectroscopy. And then using this technique, we can start with the simplest sample that's possible, which is a 3-level atom. So here we're talking about potassium atom. Since we couldn't resolve the hyperfine level in this experiment, so we treat the system as a very simple 3-level V system, where you have the ground state, which is the S state, and then two excited states, which are both p states, and these are the famous D1 and D2 transitions. So if you perform an absorption measurement, then you will have two absorption resonances, which are corresponding to D1 and D2 transitions. So now we can do 2D spectroscopy with this particular pulse sequence, which is known as the rephasing pulse sequence. And then if you do 2D spectroscopy on the sample, and this is what you get. And that's exactly what you would expect from a 3-level V system. So you have two diagonal peaks, which are corresponding to the D1 transition and D2 transition. If you remember my analogy of tuning forks, right? So now you have two tuning forks, tuning fork number 1, tuning fork number 2, and you see the resonances of tuning fork 1, tuning fork 2. And also in this case, you see the coupling between the two transitions, D1 and D2, and that gives you two cross peaks right here and right there and there. In fact, each of the cross peaks actually has two different contributions. One comes from the ground state depletion, and the other one comes from the quantum coherence between the two excited states. Okay, so that is the 2D spectrum you will get from a single atom picture. And of course, that's a very good way to actually verify that our setup actually works, and we got the results that we would expect from the simplest system. But what we were really interested in is the many-body physics in an atomic system. So what we want to see is that if you increase the number of atoms in your system, what's going to happen? What's the interaction between them, right? So that's the physics we would like to study, and we start we start with a 2-level with the simplest picture with just two atoms. Here we consider just two 2-level atoms. Each atom has a ground state 0 and excited state 1. Now, in the two-atom collective picture, we can view this two-level atom system as a 4-level system, where the ground state has two atoms in the ground state, and then the singly excited states have one atom excited and the other atom still in the ground state, but you don't know which one. And the doubly excited state has two atoms in the excited state, and these are the so-called Dicke states of two atoms, right? So now, if you introduce a little bit of interaction between the two atoms, what the interaction will do is to introduce an energy shift for both the single excited states and the doubly excited state. And also the interaction will change the dephasing rate from the doubly excited state to the singly excited state, and then from the singly excited state to the ground state. Therefore, the symmetry of this 4-level system is no longer there. It's not a symmetric system anymore. And that also gives you a way to actually detect the interaction between two atoms. And of course, this picture can then be extended to a 3-level, to a 3-atom system, which is similar, or for that matter, you can extend this picture to n atoms. Now, the question is, if there are interactions, in this case, dipole-dipole interactions between the atoms, how many atoms should be accounted in the system, and what's the interaction strength, what's the range of the interaction, and can atoms be correlated, and what's the dynamics of the interaction. So these are the questions we were interested in. Now, in order to look at the two-atom states, we need to do a slightly different experiment. So the experiment, the 2D spectroscopy experiment we do here is based on this 3-level model system, where you have a ground state 0, singly excited state 1, and then doubly excited state 2. And then we're going to use a pulse sequence to do the so-called double quantum excitation, where the first two pulses excite a quantum coherence between the ground state 0 and the doubly excited state 2. That is the superposition of the ground state 0 and state 2, right? And then the third pulse then converts that double quantum coherence back into a single quantum coherence, which can then emit as a signal. So the reason we're doing this is to generate this double quantum coherence between 0 and 2. And then this coherence actually oscillates at the frequency that is double the frequency from 0 to 1. But then the detection is actually at the single quantum frequency regime. Therefore, you don't have to detect the two photon frequency, but you will still be able to probe the double quantum coherence, which evolves during the second time delay. Therefore, if we measure the signal that emits after third pulse, and then measure that signal as a function of the second time delay, capital T, then we will be able to actually correlate the signal with whatever dynamics that's happening in the second time delay. And now if we perform a Fourier transform of that, then we will get a spectral signal which is on the diagonal line. Oh, but this is not really the diagonal line. This is actually y is equal to 2x diagonal line, because the y-axis has the double frequency as the x-axis. Now, this signal actually relies on the existence of this doubly excited state 2. All right, now back to potassium. So if you look at the two-atom picture of potassium, for single atom states, you have two states corresponding to D1 and D2 states. But the two atom states have three possible energy levels, which are the combination of D1 and D2. So now if you perform the so-called double quantum 2D spectroscopy on the system, then in the y-axis, you're going to see three resonances, which correspond to these three energy levels, which are combinations of D1s and D2. So therefore, this is a direct observation of the two atom states, two atom correlated states in the system. And also this signal of double quantum spectrum actually relies on some interaction between the two atoms in order to break the symmetry. Therefore, this is also a detection of the dipole-dipole interaction between these two atoms. But it's a very weak dipole-dipole interaction, which doesn't really shift the energy level by any measurable amount. Okay, so now that was two atoms. Now you could ask me, okay, so now what if we have three atoms together? Then in that case, in the three atom picture, the three atoms can have four different energy levels, which are again the combinations of D1 and D2 states. Now, in order to probe the three atom states, you will have to perform a much higher order nonlinear experiment, which will be able to excite the system from the ground state to the singly excited state, to the doubly excited state, and then to the triply excited state. To do that, you need more pulses. However, in the experiment, we have a trick, that is, we don't really need 6 pulses in this experiment to really excite the triply excited state. One trick is that you can always treat one pulse as 2 pulses, but with 0 time delay. Therefore, each one pulse can be used as 2 pulses. It's just they have 0 time delay between them. Or for that matter, you can treat one pulse as 3 pulses or even more pulses with 0 time delay between them. As long as you don't need to scan the time delay between those pulses. and that and we can actually do that, and then the signal associated with this higher order excitation needs to be demodulated at different frequencies, and all of that can be done by electronics and reprogramming the electronics. Okay, so that's a detail. Now we went ahead and we did the experiment in order to probe the 3 atom states, and here's what we got. And we have, for sure, we have 4 energy levels in the y-axis, which correspond to these 4 energy levels for 3 atom states. And then after that, then I asked my student, well, why don't you try even higher order measurements? And for sure, he went ahead and did the experiment with even higher order nonlinear response, and that shows the coordinated states of 4 atoms. And then we kept going to higher and higher order nonlinear response, and then we start to see the 5 atom states, and then 6 atom states, and then all the way to 8 atom states. So now what we see here is a measurement of multi-atom Dicke states from all the way from 2 atoms to 8 different atoms, and we can actually selectively, deterministically to detect what number of atoms that we would like to see. So now with that, then you can actually now study things like the quantum dynamics of those multi-atom states, and try to measure what's the decoherence time for each state. And of course, here is a very simple system, and that scales linearly with the number of atoms. But that's just an example. Now, all of the experiments I've talked so far, they are done in an atomic vapor. So that actually creates a problem, that is because it's in an atomic vapor, so the distribution of the atoms are is a statistic result. So you don't have a deterministic number of atoms and the spatial distribution of the atoms. So an ideal experiment to study how the property scales with the number of atoms would be something like this, or would be if you can actually grab just 2 atoms and arrange them in space as you like, or for this matter, you can have an array of atoms, right? And then you can perform the experiment we just talked about, and try to study how the properties of this system actually scale with the number of atoms or the geometry arrangement of the atoms. So that would be an ideal experiment. However, is really hard to actually grab an atom and arrange them in space. Until very recently, that was not possible. So in order to do that, so the first step we need to do is to actually cool down the atoms. In this case, we use laser cooling to cool down the atoms. And this is a standard magneto-optic trap mod, and we can cool the atoms down to the order of about 100 microkelvins, and then we have about 1 million atoms in the trap. And now the question is, this density and the number of atoms are different from what we saw in atomic vapor. So the question is, well, is it even possible to do 2D spectroscopy in cold atoms? And that's what we were trying to figure out first. So we actually combined a cold atom mod setup with our 2D spectroscopy setup, and then we took some initial data already, and which shows that we can get palm probe signal, and then nonlinear four-way mixing signal from the cold atom cloud, and then later we were able to perform 2D spectroscopy on the cold atom. Now the next thing would be now you have a mod, you have a bunch of cold atoms which are not moving that much in the cloud. And then what we can do is to shine focused laser beams, which are basically optic tweezers. And now each tweezer beam will be able to trap one atom or no atom. And then if the traps no atom, then you can actually destroy the trap and rearrange the trap. This way, you can have an array of atoms arranged in any pattern you would like to have. And then we combine 2D spectroscopy setup with the atom array. And now you will be able to do 2D spectroscopy an array of atoms with a deterministic number of atoms and also very specific spatial distribution of the atoms. And this is something we are still working on right now. Okay, so now we're going to switch the gear to semiconductor optics. So normally I start with this slide for semiconductor optics, but I don't think this audience requires any introduction for that. So let's say if we look at, if we are looking at gallium arsenide bulk, then the laser can, or light can excite different carriers in the sample. You can form then excitons or other quasi particles in the sample, and all of these quasi particles can actually interact with each other. So in my view, a semiconductor intrinsically is a many-body problem, right? So now it turns out 2D spectroscopy has been used to study those many-body dynamics, carrier dynamics in different semiconductor materials, including quantum wells, quantum dots, 2D materials, and perovskite materials. And this is just a very incomplete list of some of the examples. Now back to 2D materials, and you have a similar situation. You have all of these quasi particles, excitons, biexcitons, trions, polarons, and they actually can interact with each other through quantum interaction, and therefore you're actually looking at a very complicated many-body system. Then it's very interesting to actually probe now what are the dynamics, and the dynamics can be quite complicated in this system. And it's very interesting to actually study this system with all of these different quasi-particles on one hand. This provides a very nice playground to actually test different models of many-body physics, especially in the context of a 2D confinement. But on the other hand, it's important for applications. It's important to understand properties of the quantum dynamics of all of these quasi-particles. So besides the population relaxation processes, it's also important, especially for quantum applications, to understand the quantum coherence in the system, including inter-valley and inter-valley coherence. And it's essential to actually be able to have a tool to directly probe quantum coherence and measure its dynamics, especially the decoherence time or T2 time associated with this quantum coherence. So now what 2D spectroscopy can do is to provide you a way to directly measure those dynamics. For example, you can do the so-called rephasing 2D spectroscopy, where if you have a sigma plus polarization light interacting with a k-valley, and that generates an inter-valley coherence, and then you can actually measure the coherence time of that. Now, when you do that, one problem is the inhomogeneous broadening, right? So now 2D spectroscopy actually has this advantage of being able to separate homogeneous and inhomogeneous linewidths. So the idea is that if you have a homogeneous system in 2D spectrum, the signal is actually symmetric in both directions, in the diagonal and the cross diagonal direction. Therefore, the line width in this case actually corresponds to the homogeneous line width. Now, if the system is actually inhomogeneously broadened, then what you have is that the 2D spectrum is actually elongated in one direction, but not the other direction. So you can sort of think of now you have an inhomogeneously broadened system of many different oscillators. Then what's going to happen is that each oscillator has a star-shaped peak on the diagonal line, but now you're going to distribute all of these oscillators along the diagonal line. So that's why this spectrum is actually elongated in the diagonal line. But then the other direction, the cross-diagonal direction, will still have the information about the homogeneous line width. Therefore, we can actually analyze the line width in both directions in the homogeneous, in the cross-diagonal direction and the diagonal direction. Then we actually figure out a way to actually extract both the inhomogeneous line width and the homogeneous line width in the system, even with the presence of very strong inhomogeneous broadening in the system. So, for example, we did this for a MOSE2 monolayer, and this is a rephasing 2D spectrum of trions in MOSE2 monolayer, and this spectrum is slightly elongated in the diagonal direction, and then we can do a line width analysis in the cross diagonal direction, and that allows you to figure out the homogeneous line width in that direction. Now, if we do this with different power, then you can actually see the excitation-induced dephasing effect as you increase the laser power. So the line width actually changes when the laser power increases. But we can actually extract the homogeneous line width at different laser powers. And then if we plot them, then you can see how this actually changes with the laser power. Then what we can do is to fit this with a straight line so that we can actually extrapolate the homogeneous line width at the zero excitation density or at the zero laser power, right? And then that is done at 5 kelvins. And then we can do the same thing, extract the zero power homogeneous line width at different temperatures. And then by extrapolating from this data set, then you will be able to find out the intrinsic homogeneous line width at the zero excitation power and at zero temperature, which will be quite useful for understanding this sample. And it turns out that line width is quite broad, and that corresponds to a decoherence time of about 200 femtoseconds, which is really, really fast. Now, if you think about this, the T1 time for this sample is about at the picosecond scale. So this decoherence time is much shorter than the population lifetime, which tells you some important information about the decoherence processes in the system. Okay, so now if you use different polarizations, then you can actually selectively excite different values. So for example, if you use sigma plus 2 pulses, then you will generate a population in the K value. And then there could be an exchange between K and K prime. And then if that happens, and then the third pulse is sigma minus, then you will be able to probe the signal that comes from the other value, which actually is dependent on this exchange between the two. Therefore, by doing these experiments, you can actually directly probe the value exchange between the two values. And this is an example of the 2D spectrum from that measurement. And this is a direct probe of the fatty exchange between the two. Now you can do a series of measurement like this with different capital T delays that will tell you the time dynamics of that process. And you can also do sigma plus, sigma minus, and sigma plus. In this case, you will be able to generate a fatty coherence, inter-fatty coherence between K prime. And if you wait for that to evolve and then probe it with the third pulse, then that will give you the dynamics of the inter-fatty coherence as a function of the waiting time right here. Also, that's a direct probe of that quantum coherence. Okay, so so I will finish in 5 minutes. And just one last example. More recently, we started looking at more complex quasi-particles. This is something known as Fermi polarons, and what this is basically the excitons that are coherently dressed by the surrounding Fermi sea. And then the dressing results in two different solutions. First, the attractive interaction between excitons and Fermi sea leads to the so-called attractive polaron, which is energetically favorable. It actually has a lower energy and therefore is more stable. And then also we get a higher energy repulsive polaron, which is a metastable state, and eventually decays back into the attractive polaron. So normally it's difficult to distinguish the trion picture and the polaron picture at really low doping density when the Fermi energy is smaller than the intrinsic exciton linewidth. So what we could do is to actually increase the doping level. Therefore, you will see two branches of resonances emerge from, in this case, is the reflection spectroscopy measurement. You will see this attractive polaron branch and the repulsive polaron branch. Therefore, another question we ask here is that, okay, so what about the quantum coherence dynamics? Is there any difference between the attractive polaron and the repulsive polaron branches? So what we did here is that using this sample, we can adjust the doping level, and then we perform 2D spectroscopy at different doping levels. At the 0 volt gate voltage, the spectrum shows only one peak, and that peak comes from the exciton resonance. And then we can, of course, perform the linewidth analysis in order to extract the homogeneous linewidth. And then as we increase the gate voltage, there now have two peaks on the diagonal line, and they are corresponding to the attractive and repulsive polarons. And also you'll see the two cross peaks, which are corresponding to the coupling between these two branches. And then at 1.3 volts, you only see the attractive polarons, and the repulsive polarons is no longer there. However, you still see this little bit of cross peak, and that suggests that what happened to the repulsive polaron is that the repulsive polarons actually was excited initially, but very quickly decays back into the attractive polarons within the pulse width about 100 femtoseconds. And then at even higher voltage, the attractive polaron starts to get really broad and shows a broader homogeneous linewidth, which corresponds to faster dynamics. And then that state becomes unstable. And you can clearly see the dynamics as you change. You can clearly see the different resonances coming from different quasi-particles as you change the gate voltage. and also by analyzing the linewidth for each resonance, then you will be able to extract the dynamics of each, right? So we did this. This is more quantitative analysis of the linewidth as well as the resonant frequency positions, and then we compare this in collaboration with our series. We can compare this to the polaron theory, and then the experiment matches pretty well with the theory. Okay, so I think my time is almost up. With this, I'd like to just to summarize that that this technique of 2D spectroscopy or multidimensional coherent spectroscopy is a very good tool to probe matter-body interaction and correlation in different systems, in the atomic systems, as well as the 2D materials. And in the future, we will be continuing in this line of research to look at even more details of the dynamics in both the atomic systems and different other semiconductor materials. So so I will stop here. With that, I would like to thank you for your attention, and if you would like to learn more about 2D spectroscopy, I will just put this last slide on the screen. And thank you, and I'm open for your questions.
Thank you, Dr. Lee. Does anyone online or in the room have a question?
Yeah. Thank you very much for the presentation. I was, you know, the ability to measure dipole-dipole interaction was curious to me, but I didn't know if that's something that you've explored in the semiconductor systems that you looked at. You were showing that for your multi-state atom systems. Is that something you've explored in, like, maybe potentially heterogeneous semiconductors or like quantum dots or any of the other systems? Has that been explored?
So thank you for your question. This is right to the point. This is something that we are interested in. So normally in a semiconductor system, so for example in gallium arsenide, so quantum wells, so you always have quantum interactions. These are much stronger interaction than dipole-dipole interaction, and the system is way more complicated to study. So what we've been thinking about, let me see if I have a backup slide. So okay, so we've been thinking about something like this, right? So if you have an array of quantum dots, then you can actually look at something similar. So this dipole-dipole interaction between the quantum dots, and you do this in an array. And another system we're looking at right now is an array of color centers in diamonds. I don't have a slide for that, but we were able to, in collaboration with Sandia, we were able to fabricate a sample where you can very precisely implant with ion implantation, exactly one color center at a precise location. Therefore, you can actually make an array of color centers. And then we're actually looking, we're trying to look at the interaction or matter-body correlation within that system. Yeah.
Any other questions? Okay, well, thank you so much, Dr. Lee.
Yeah.
One more.
Yeah, I was gonna. What would you say is the greatest challenge with this technique? I mean, I imagine. I mean, you're set up for the, you know, like the Mott with a, you know, you had a microscope and now you're coupling your pulse lasers. I just imagine these measurements must take months. Is that like, what is the level of difficulty for these, you know, these measurements that you're doing?
And you are absolutely right. These are, so especially the one you just mentioned, combining 2D spectroscopy with the cold atom setup, that's actually really, really complex. So but I think the technique 2D spectroscopy, this technique becomes more user friendly as the time goes. So if we go back 10 years, I think only a few very specialized groups that can actually perform this type of measurements. But now they're actually commercially available setup. I mean, for doing 2D spectroscopy. So you can actually buy commercial setup for that. For example, there's one company at University of Michigan called Monster Sense. They can provide you this setup, and it's much more user friendly right now. And my hope is that that a lot more people can start using this technique. Yeah, so just looking back how MR was developed, right? So now you can do an MRI imaging scan without knowing what's actually in the MR machine. I think we have the potential to go into that direction as well.
Thanks for sharing.
Thank you.
Thank you everyone for your attendance, and thank you, Dr. Lee.
Yeah, thank you.
Thanks, bye.
Bye.